layer chromatography, I just have to plug how much our things cost in comparison to university methods. We can actually chromatographically resolve the A to E and show that it's really it that's going away, that the bleaching is not all that we have. We can actually show that this is due to A to E degradation. And our friends at the universities confirm that with their high-tech gear, of course, that produces these pretty curves. And this is just a close-up of the HPLC degradation over time, showing various peroxidase type enzymes that we found, which also efficiently destroy A to E. And these enzymes destroy it completely. They just oxidize all the double bonds. But they're non-selective, so they have a higher potential for side effects. And we don't know yet which is better, complete molecular destruction that we see from the peroxidase or the selective scalpel-like cleavage that I showed you earlier from the carotenoid oxygenase. One very important thing is the phototoxicity of A to E is mediated by epoxide formation, you can see here. These are highly reactive, react with DNA and all kinds of things. So we were delighted to see that when we had photodegradation products of A to E and compared them with our enzymatic products, our products were very different. And these stain with aldehyde selective dyes. So these are aldehyde products. The side chains just get successively shortened until small molecule aldehydes are left. Tests in human tissue culture. This is how you guys do tissue culture, right? We have our own model here. This is an air purifier from Walmart that's blowing into a plastic box, and we do cell culture down here. And it works just fine. We never had any problems with contamination. There are also some UV lights on the inside for sterilization. But I'm going to show you data from our collaborator, who is at Columbia University, who luckily completely took over the preclinical testing of these enzymes on her own dime now. So that was really the goal, that we could just do on the cheap the initial milestone and then get academic collaborators interested in using their resources to do the main parts of the work. So this is a cell membrane antibody that delineates the cell boundaries in red, and the green stuff is A to E loaded in lysosomes fluorescing there. So it's convenient if you have a fluorescent substrate, even for your high-tech gear, you can just see how much of it is there. She used our enzyme, successfully delivered it into lysosomes. So you see horseradish peroxidase is one of our peroxidases, lysotracker, they are together. So it means our enzyme got into the lysosomes. And she just used a protein transduction reagent that's commercially available. So it would probably not work in vivo this way. This is just a cell culture reagent that she used to get them there. And she brought the A to E content down by something like a third. For comparison, if you bring the A to E content down by a third using light, the cells will roll up and die. You produce way too many epoxides and free radicals. So our enzymes non-toxically achieve this amount of degradation for the first time. And the enzyme treatment also was not toxic in her hands. So that's where we are with macular degeneration. Now what we really need to do is implement a drug delivery system that will work in vivo, such as the one Jeff introduced us to this morning, the guild lysosomal targeting, or the mannose-6-phosphate-based lysosomal targeting. And that's what the main occupation of our lab is now, really, to construct these drug delivery systems. Second project that we have is atherosclerosis, and I'm going to take you through that a lot more briefly. And this is highly simplifying. In atherosclerosis, this is a clogged artery. Blood would be flowing like out of the plane that we're seeing here in a very narrow volume. And highly simplified, outrageously simplified. If you have macrophages, you can get them in a dish to process cholesterol deposits. But if you poison them with a little bit of this guy, 7-ketocholesterol, they turn into these atherosclerotic foam cells. So we think that the storage problem in atherosclerosis is not cholesterol, because the macrophages know how to deal with it, but it's really 7-ketocholesterol, the poison that selectively compromises the macrophage's cholesterol processing ability. So we cultured microbes from around the world, looking for the ability to biodegrade 7-ketocholesterol. Again, university model of the shaker versus our own. You can see our styrofoam box that's being powered by a household fan that's getting cooling from a second fan. Works just fine. Specifications, RPM, temperature accuracy is all the same. These are the degradation curves we recorded from various soil isolates. This again HPLC data or thin layer chromatographies. They tell us the story, they're just not as pretty for a presentation. And we found the enzyme that these microbes use, pretty much all of them, is cholesterol oxidase. That makes oxidase as this group. And so we were wondering, maybe this little modification, it looks small, it's not really a degradation, it's just a little modification. Is that enough to prevent the toxicity of the compound? So we poisoned cells, human cultured cells, in our cell culture apparatus with 7-ketocholesterol, and you see cholesterol is not very toxic to them at all. 7-ketocholesterol is highly toxic, and this is the product that our enzyme makes, so it's intermediate. So not as good as we had hoped, but still it's some prevention of toxicity. And other people have discovered a whole bunch of 7-ketocholesterol degrading enzymes in the meantime. Some of them discovered by us, others by other groups. And now we're, again, trying to implement a drug delivery system to use all these 7-ketocholesterol enzymes, which are now known, get them all into human cell lysosomes, and see if we can do anything about the storage problem in vivo. And then another shameless plug for 2 other SENS Foundation Research Centre scientists who are presenting at this conference. Justin Revo will talk about his senescent T lymphocyte removal project, where he uses magnetic antibodies to remove the senescent T cells from the blood, giving the young cells space to repopulate the body. Saturday, 5 p.m. And this is Nathan Schuler, who will be talking about his lipofuscin-destroying lasers. Thank you all for coming. That's it.